Good day, everyone. Today we will be having our final journal review for this semester. And our journal review for today is all about phytohormones. And the title of the journal that we will be tackled for this day is a novel Rice or Rhiza sativa L, acidic PR1 gene highly responsive to cut, phytohormones, and protein phosphatase inhibitors. Presented by yours truly, Gilbert L. Florage. And here are the respective authors of this journal: Ganesh Kumar Agrawal, Nam Sojwa, and Randep Rakwal. And now let us proceed to the introduction. Interest in rice is an important socio-economic crop in Southeast Asia. It's focused on the rice plant self-defense mechanism, which started with identification of defense-related and cellular protectant proteins from stressed rice plants. The HR response characterizing plant pathogen interactions is not only associated with synthesis of low molecular weight secondary metabolites. Example is toxic phytoalexins, cell wall rigidification as a result of callose, lignin, and suberin deposition, but also with the induction of a variety of novel proteins, collectively referred to as the pathogenesis-related proteins, or PRS. It has often been suggested that the collective set of PRS may be effective in inhibiting pathogen growth, multiplication, and/or spread, and be responsible for the state of systematic acquired resistance. Moreover, the induction of PRS has been found in many plant species belonging to various families, suggestive of a general role for these proteins in adaptation to biotic stress conditions. Among these PRS, the PR1 class is a dominant group that is induced by pathogens and is commonly used as a marker for SAR, with only the PR1 members from dicots like tobacco and tomato being well characterized. The importance of the phytohormones salicylic acid and jasmonic acid as a critical signals in induced resistance responses in plants is recognized. In previous studies of jasmonic acid affecting rice secondary metabolite production and induction of a variety of defense-related proteins, provide additional evidence for the importance of jasmonic acid in rice plant self-defense. Though the pathways of a jasmonic acid action and its interaction with other phytohormones remain to be clarified. Although jasmonic acid was shown to induce some members of the PRS in rice, we were not available to identify any proteins with homology to the PR1 protein family. Considering the importance of PR1 family of proteins in plant self-defense and the role of a jasmonic acid as a signaling molecule in rice plants, it was reasoned that jasmonic acid should also have an ability to affect expression of the PR1 genes in rice. Thus, in this study, planned and conducted these present experiments with a defined aim in mind to identify jasmonic acid-induced rice PR1 genes. So before we proceed to methodology, let me describe the cloning and characterization of a novel acidic PR1 gene from jasmonic acid-treated rice seedling leaf, which designated as OSPR1A. This OSPR1A gene comprises a multi-gene family in rice and encodes a highly acidic low molecular weight protein of PI 4.4. Other than jasmonic acid inducibility, this OSPR1A gene was found to be highly responsive to cutting, exogenous application of other phytohormones, and protein phosphatase inhibitors. And furthermore, cytoplasmic or de novo protein synthesis was shown to be required for OSPR1A gene activation by these compounds. But on the other hand, accumulation of OSPR1A transcript was enhanced by using a potent protein kinase inhibitor or straurosporin in other term. Simultaneously with protein phosphatase inhibitors but not with jasmonic acid, it results presented in this study indicate that OSPR1A gene expression is regulated by an interaction of phytohormones with light signals, which is more likely mediated via a protein phosphorylation. The phosphorylation events in other side. But this is the first report on cloning of a novel acidic PR1 gene member from rice. So, as I've discussed to you, this scientific journal is the first report on cloning of a novel acidic PR1 gene member from rice. Now, here in methodology, I will be discussing the plant materials used, the chemicals, the construction and screening of rice, DNA library, DNA sequencing and sequence analysis, the northern blot analysis, and the southern blot analysis. The plant materials used in this study is obviously rice, with a variety of Nippon Bare. Seedlings cultivated in the greenhouse at 25 degrees Celsius, 70% relative humidity, and a 12 light period day. At 14-day-old, the fully expanded leaves were used as the experimental material. Approximately 2 cm long leaf segments were cut with clean scissors and floated on solutions. 10 ml is the final volume of the solution, and the test compounds in sterile Petri dishes. For observing the direct effect of ethylene gas, rice leaf segments were floated on millicue water in Petri dishes containing a 2.5 ml solution of ET in a small 15 ml Falcon tube cap. There was no direct physical contact between either the two solutions or the ET solution and rice leaf segment. Millicue water-treated leaf was used as control. While the incubation was done under continuous light or under darkness, as indicated in the figure legends, leaf was harvested at the times indicated and used immediately or stored at 280 degrees Celsius till total RNA was extracted. Next is the chemicals used by this study. First is the cantharidin or CN. Next is JA with the racemic mixture and SA. SA were purchased from Sigma St. Louis, and CN was prepared in acetone as a 100 ml stock solution, whereas jasmonic acid was dissolved in methanol as a stock solution of 100 ml and used as concentrations. Endothal was obtained from Biomol Research Laboratories Incorporations and used from a stock solution of 100 ml in millicue water. The protein synthesis inhibitors or CHX and tetracycline or TET were from Sigma and used as 10 ml concentrations. The phytohormones are abscisic acid, ABA, kinetin or KN, gibberellin or GA3, and 3-indolleacetic acid or IAA were obtained from Wako Pure Chemicals, Tokyo, Japan, and used at 100 ml concentrations. ST, a potent serine threonine protein kinase inhibitor, was from Sigma and dissolved in DMSO as a 100 ml stock solution. All other chemicals used in this study were of analytical grade. Next is the construction and screening of a rice DNA library. Poly A and RNA, which is 5 mg, was prepared from rice leaves which had been treated with 100 ml jasmonic acid for 24 hours and used for construction of a DNA library. The library was constructed in 1-ZAP Strategene Lahola CA according to the manufacturer's instruction. To obtain a hybridization probe, a rice DNA fragment corresponding to the maize acidic PR1 gene was applied by PCR on the basis of maize nucleotide sequence published previously. Accession number of U82200 database General Bank. Sense and antisense degenerate primers used for PCR were as follows. As we can see in consensus 1, that is the DNA screening of the rice, and the another is the consensus 2. So, as being flashed in the screen. The first strand DNA synthesized using 10 mg total RNA isolated from 100 ml jasmonic acid. Treated rice seedling leaf for 24 hours under light with a StartaScript RT-PCR kit. Strategene, following the manufacturer's protocol, was used as a template. The resultant PCR product of 321BP was ligated into a PGEM T vector or Promega Madison WI and sequenced. The PCR product was labeled with DCTP by the Multiprime DNA Labeling System, Amersham, Buckinghamshire, UK, and they used as a probe for screening the DNA library. PageMed vectors or PBLUscript SK were excised from selected positive clones using in vivo excision system or Strategene. In DNA sequencing and sequence analysis method, plasmids were isolated according to the standard protocol and sequenced using a determinator cycle sequencing kit or Perkin-Elmer or Foster CTSA. All the sequencing data were analyzed using genetic software, which is SDC Software Development, Tokyo, Japan. Searches for information and homology of nucleotide and amino acid sequence was analyzed using homology search with BLAST against the sequences in the GenBank and EMBL DNA database. Next is the Northern blot analysis method. RNA was isolated from rice seedling leaf using the RNA plant kit as per the protocol provided by the manufacturer. Total RNA is 20 mg was separated on a 1.2% formaldehyde denaturing agarose gel as described. Blot onto a nylon membrane or high-bond nitrogen-1 amersham and hybridized with a 32P labeled PCR amplified partial OSPR1A DNA probe. It was used a 602 BP using a primer pairs of OSPR1AF and OSPR1AR as being showed in the screen, using a multi-prime DNA labeling system by Amersham. Hybridization was carried out as described previously. Equal loading was confirmed by iridium bromide staining, or after blotting by staining of RNA with methylene blue. The membrane was washed with 23 SSC and 0.1% SDS at 65 degrees Celsius for 1 hour and exposed to an X-ray film, which is from Kodak, Tokyo, Japan, for 18 hours at 280 degrees Celsius. And about the Southern blot analysis method, rice genomic DNA, which has 100 mg from rice leaves, was digested with Hindi third, Echo R1, XBAL, BAM-HI or PSTI, separated by electrophoresis on a 0.8% agarose gel and blotted onto a nylon membrane by a alkaline transferred method. The membrane was hybridized with 32P labeled PCR applied OSPR1A partial cDNA probe, and as we can see, the OSPR1A F1 gene and the OSPR1A R1 gene, washed with 23 SSC and 0.1% SDS at 65 degrees Celsius for 1 hour, followed by an additional washing with 0.23% SSC and 0.1% SDC at 65 degrees Celsius for 30 minutes and exposed to an X-ray film for 18 hours at 200 degrees Celsius. And now let us proceed to results and discussion. First is the isolation of a rice acidic PR1 DNA from jasmonic acid treated seedling leaf. As our primary interest was in cloning the PR1 gene from jasmonic acid treated rice, especially the acidic PR1 genes, of which there is no report to date, we used the previously published MACE acidic PR1 gene sequence to design the generated primer pairs and carried out RT-PCR using total RNA extracted from 24-hour jasmonic acid treated rice seedling leaf. A PCR amplified by 3:1 BPC DNA fragment was isolated from agarose gel sequence, and the nucleotide sequence was found to be highly homologous to the MACE acidic PR1 gene sequence. With this information in hand, it proceeded with the isolation of a full-length DNA clone by DNA library screening. As being showed in the figure, to isolate a full-length rice acidic PR1 DNA, a DNA library of jasmonic acid-treated rice seedling leaf was prepared in 1-ZAP2 strategy. With the above homologous DNA fragment as a probe, a total of 20 positive clones of various lengths were obtained after screening of 200,000 recombinant clones of the DNA library using plaque hybridization. The DNA inserts were isolated from the blue script plasmids in the selected DNA clones and digested with Echo R1. So first, and the size of the DNA inserts was determined by agarose gel electrophoresis. The putative full-length fragment were partially sequenced. The sequence comparisons of the DNA clones by the BLAST program indicated that the 3 DNA clones of abundant 0.08, as you can see in the figure, are 100% identical to one another in nucleotides and encode an acidic PR1 protein. One of the 3 rise acidic PR1 DNA designed as OSPR1A was used for further analysis. Next figure is the OSPR1A DNA sequence. Is 830 BP long and contains an open reading frame of 507 nucleotides, beginning with the first ATG initiation codon at nucleotide position 99 and ending with a TGA termination codon at position 606. The OSPR1A possesses a putative TA box beginning at the nucleotide position of 44 and 40A repeat sequence at the top hatch underlines, beginning as a potential target site for transacting factors in plants. This indicates that the presence of putative box or the TATA box just upstream of the TSP in the OSPR1 leader sequence. It might serve as a CIS element for as yet unidentified transacting factors regulating OSPR1 gene expression. The presence of 4 TATA repeat sequences between the putative box and TSP in the OSPR1 leader sequence was not found during a survey of at least the PR1 genes reported in the database. It is speculated that these repeated sequences might serve as an additional CIS acting elements. A careful observation of nucleotide sequence upstream of the TSP was revealed the presence of a CAT box or the CAAT box. Next is the genomic organization of the OSPR1A. To determine the copy number of OSPR1A in the genome of rice, it performs southern hybridization as a probe. As mentioned in the materials and methods, multiple distinct hybridizing bands were detected with the 4 restriction enzyme, which is HinD3 and ECO-R1, XBAI, and BAM-HI. This result clearly indicates that OSPR1A belongs to a multi-gene family in rice. So that is the result of the genomic organization of the OSPR1. It result clearly indicates that OSPR1A belongs to a multi-gene family in rice. This result is also similar with maize, where based on gene sequence homology and southern blot analysis, a maize PR1A acidic gene family with multiple members is proposed. So as being shown in the figure, it is a genomic southern blot analysis of OSPR1A. Rice genomic DNA with a 1 milligram from rice leaves was digested with HinD3 ECO-R1 XBAI BAM-HI or PSTI, separated by electrophoresis on a 0.8% agarose gel and blotted onto a nylon membrane, high bond N, by Amersham. The membrane was hybridized with a P-labeled PCR applied of OSPR1A partial DNA probe as mentioned under materials and methods. The sizes of the molecular weight marker are indicated on the left-hand side. As being shown in the figure, there is no report on induction of an acidic PR1 gene by jasmonics, thereby presenting first evidence in support of J or jasmonic acid as an effective inducer of, and at least, an acidic member of PR1 gene family in rice. Interestingly, a time-dependent accumulation of this OSPR1A transcript was also seen in the water controls, indicating a highly cut-sensitive gene expression. However, the transcript leaves were relatively lower than that observed after jasmonic application. And again, there are no reports on Q-inducible acidic PR1 genes in monocots or dicots, though a recent report shows that mock or water-inoculated maize leaf showed a weak accumulation of the acidic PR1 transcript, that though a 14-day after treatment. Our next figure shows the results indicate that the requirement for protein synthesis precedes activation of the OSPR1 gene, and that this de novo protein synthesis is cytoplasmic in nature. Cycloheximide blocks OSPR1 gene expression, whereas staurosporine enhances its induction with CN or EN. A northern blot analysis of RNA from leaf treated with 100 ml jasmonic acid or 1 ml of CN and EN alone. Simultaneously, with 10 ml of CHX or 10 ml of TET as indicated on the top of each lane, as we can see in the figure, the water control is shown in lane 1. But in northern blot analysis of RNA from leaf treated with 100 ml, it's simultaneously treatment with 10 ml of staurosporine. The effects of 10 ml ST on CN and EN induced of, as we can see, there is an induced of OSPR1 transcript levels as being shown in lanes of 3 to 6 of the induced PR1 transcript levels. The total RNA was extracted at the 24-hour after treatment under continuous light. But as we can see, there is an equal loading of 20 milligram and confirmed after blotting by staining of RNA with the methylene blue. So as we can see in the figure, that is the stain of the RNA with the methylene blue, and blots were hybridized to the radio-labeled of OSPR1A PCR product. The next figure showed the examinations of the effect of some of these phytohormones such as jasmonic acid inducible on PSPR1A transcript levels. Result that the JA or jasmonic acid induced accumulation on OSPR1 transcript was almost blocked by a simultaneous treatment with 100 ml SA, as being showed in the lane 8, the ABA, which is in the lane 10, there is a higher concentration of 200 ml of jasmonic and SA acid. The inhibition was complete. The corresponding of relative RNA abundance in percentage is given in a figure as being shown. The result provides the first evidence of exogenous SA blocking jasmonic acid inducible PR gene. So this, as we can see in the figure, this is the first evidence of the exogenous of blocking the SA in the jasmonic acid inducible in PR gene. And as we can see, there is an activation in monocots, which is the rice, thereby suggesting similarities in the mechanism by a SA might block jasmonic acid inducible defense gene among dicots and monocots. In addition, it shows that it is not only in SA acid but also ABA was equally effective blocking the jasmonic acid inducible in OSPR1A gene expression. A first report, but on the other hand, KN was also showing some inhibitory effect on the accumulation of the OSPR1A transcript when applied simultaneously with jasmonic acid. These results indicate to a complexity in the crosstalk between phytohormones and regulating defense gene activation in plants. And more studies will be needed to generalize among the signal pathway crosstalk for plant. It needs a lot more studies to examine and generalize the signal pathways across talk for plants. And now we're down to our summary. And a novel rice acidic pathogenesis related PR class 1 DNA OSPR1A was isolated from jasmonic acid treated rice seedling leaf. The OSPR1A DNA is 830 BP long and contains an open reading frame of 507 nucleotides encoding 168 amino acid residues with a predicted molecular mass of 17,560 and pi of 4.4. The deduced amino acid sequence of OSPR1A has a high level of identity with acidic and basis PR1 proteins from plants. Southern analysis revealed that OSPR1A is a member of a multi-gene family. The OSPR1A gene was found to be cut-inducible, whereas the phytohormones, jasmonic acid, and cyclic acid is 3-indoleacetic acid, gibberellin, and ethylene enhance accumulation of OSPR1A transcript as well as the protein phosphatase inhibitors cantharidin and endothal. So that's all for our final scientific journal review. Thank you and God bless.